So I'm going to talk about the origin of individuality as biologists are interested in it. It's linked to the idea of the major transitions in evolution. So let's go into business. It's somewhat surprising that the basic idea of evolution by natural selection can be summarized on one slide, but it can be. Of course, to validate it, it takes many pages, and if you face a concrete problem, then of course it can easily become rather involved. But it's fair to say, and I think Darwin would agree with it, that in order for evolution by natural selection to take place, you need units that make up a population, and those units must have a certain number of criteria. They should multiply, they should have heredity, which means that like gives rise to like. They must have variability, which simply means that heredity is not exact. Now, if it's true that if the units have at least some hereditary traits that affect either the survival and or the reproduction of those units, then in such a population, evolution by natural selection can go on. Now, there are two things to be said here. One is that this is a tacit algorithm, right? And it actually can be implemented in genetic and evolution algorithms, but it's tacit and verbal at this level, so there are quantitative issues here. For example, just to mention one, heredity must be enough accurate, because otherwise the heredity information just melts away. That's kind of a quantitative constraint. On the figure itself, you can actually see that there are two kinds of entities here, A and B, and B obviously multiplies more efficiently than A, and C is the newcomer, which wasn't there. That's thanks to variability. And the third interesting thing is that this approach does not restrict it to genes or organisms, so any kind of units that satisfy those criteria can show kind of Darwinian behavior, be it molecules or some cultural items and so on. It's also true that these units of evolution are not necessarily organisms, right? That's also important. We will come back to that. What I'm going to consider here is, the very first part of the original list of the major transitions, whereby you go from replicating molecules to population of molecules in protocells. There is another one which is linked to this, that independently replicating genes can link up to chromosomes, and that's also important, but I'm not going to address it here. If you look at the whole list, there are certain recurrent themes. Novel inheritance systems arise, higher level units formed from lower level ones, and there is this phenomenon of division of labor / combination of functions that come up again and again, and that's the kind of comparative transitionology, if you wish. I'm going to start with the strictly molecular evolutionary units, naked replicators, as it were. And originally, I assumed that in terms of population genetics, there is no population structure whatsoever. So let's see. There is a very concrete problem, and from this point of departure, you can appreciate the rest. So in 1971, the Nobelist Manfred Eigen produced a landmark paper in the Naturwissenschaften, and he pointed out that It's a flip side of the coin of what the population geneticists knew, and they called it the mutation load. But Eigen also showed that given a certain fidelity of replication, which means 1 minus the mutation rate, the amount of genetic information is constrained. So if it becomes longer, or if you want it to become longer, it's not anymore sustainable by natural selection. The hereditary information melts away. This is one kind of model where you have a population of replicators, and one has a higher replication rate, let us say 10, all the others have a replication rate of 1. So the selective advantage seems to be extremely high. I mean, it's a tenfold advantage. Now, however, you can solve the system for equilibrium at various mutation rates, and here is the frequency of the different types, and here the mutation rate per digit, per bit, is increasing. And these are the equilibrium values of the different mutant classes. This is coming from here, which is unity at the top, right? This is actually the master sequence and all the other inferior competitors. And you see, this is the transition. This is what is called the Eigenian error threshold. Now, where the threshold is, of course, depends on the accuracy of replication. And in early times, it could not have been very high. I mean, that was the problem Manfred Eigen realized, and he said something that it's unlikely that a single molecular replicator, such as a piece of RNA, could have been longer than about 100 nucleotides, which is roughly the size of the transfer RNA molecule. Now, one such molecule does not make a genome. So what can you do about this? If you cannot have linked genes or linked replicators, then you must have some kind of a mechanism for coexistence. So this was a basic problem in 1971. And that was his solution also from 1971. It's the molecular hypercycle. It's basically a model of molecular mutualism, so to speak. Each member in the cycle is a replicator. They are autocatalytic, but there is also a heterocatalytic aid. So, as the arrows say, that the one has the replication of 2, 2 has the replication of 3, and 3 has the replication of 4, and you have a higher level cycle, as it were. This is by it is a hypercycle. And for the mathematically minded, because this arrow touches this arrow, the kinetics of the replication is second order. The concentration of this guy has to be multiplied by the concentration of this guy and the replication rate constant. So the molecular hypercycle from that point of view is at least second order. As it is portrayed, it's an ecological system, right? And the question is whether it's evolutionarily stable or not. And that's the problem. As it has been conceived, it's not. So there was a second series of papers in 77, and then a book was published in 79, and that book was then reviewed by John Maynard Smith. And then when he was diving into the hypercycle problem, he said, okay, wait a minute, I don't understand this. So what did John not understand? Meaning that he saw a flaw in the whole idea. And it's very simple. If you don't forget about mutations, then all kinds of problems can arise. Okay, so this is a nasty mutant. It's a mutant of I sub 1, is the P sub 1. It's a parasite. Now, what does it do? It accepts the help by the number 4 replicator, but it doesn't give anything back to the system. There is another possibility, the so-called short circuit mutant, that's also harmful, but let us consider now the parasites. So what you can prove is that if this arrow is thicker than this arrow here, in a homogeneous, well-stirred chemical reactor, and that was the original assumption by Egan, the whole system goes down the drain. The parasite just kills the hypercycle, but since the parasite cannot live without a hypercycle, and there is, for example, some degradation rate, everybody dies out in the end. So something has to be done about it. It's not going to work as ordinary chemistry works, when it is well stirred. Now we will come to a few of the resolutions, and the first types of resolution belong to multi-level selection type 1, as people call it in evolutionary biology. And the definition is actually very simple. With this kind of multi-level selection, although neighborhood effects, as you will see, are very important for the outcome of the dynamics, but only molecules replicate here. Groups do not reproduce. That's basically the definition of multi-level selection type 1. In other words, the only units of evolution are still the molecules, despite their interactions, and groups are not evolution units. That will come later. Okay, so it has been realized that many mineral surfaces are a poor man's form of compartmentation, if you wish. You can have a passive form of localization, for example, on a mineral surface, and with a limited diffusion rate in 2 dimensions. I mean, on the surface itself, and this has been shown to have important effects. One of it is the thermodynamic effect. For example, if you are interested in polymerization, where you have a leaving group, for example, water molecule, then of course it helps if the surface binds to the monomers. It binds the dimer even more, and it binds the water molecule less than the building blocks. It's a thermodynamic effect. There's a kinetic effect because the reaction rate can be boosted by the surface as an inorganic catalyst, and in a way enzymes are also surface catalysts today. The question is how general and diverse are or can be these effects. Once Leslie Orgel said that this kind of help or solution is good for polymerization, not good for metabolism. I don't fully agree with it, but we shall see some examples what can be done here. Maybe the combination of two can be very rewarding. Now, here is a model from the year 2000, and that was created in order to show that if you are on a surface, the game has been changed completely. So even for a system that would go extinct in a well-stirred reactor, you can demonstrate that stable coexistence is possible. And that's the surface metabolism catalyzed by replicator model. And in this system, there are a number of replicators here, portrayed as RNA secondary strands. Here is the metabolism. We don't detail that in this model, or don't detail it yet. And there can also be parasites. This is not shown on the figure, but of course, they can always arise, and we shouldn't forget about them, as we had just learned on the example of the hypercycle. So what are these replicators doing? These replicators can catalyze. These are the arrows, the colored arrows. They can catalyze metabolism that also produces the monomers for the replication of these molecular replicators, and this is the empty arrows. These show actually the strengths of directions whereby the replicators reap the benefits of the commonly catalyzed metabolism. Now, it's known that although this system seems to be very tightly linked with all kinds of important interactions, in a well-stirred reaction, you again have a parasite problem, because imagine that a parasite, noted here, arises which doesn't have an arrow linking from the replicator to metabolism, but only it takes the benefits. The expectation is that it can have a simpler structure because it doesn't have to perform an enzymatic function on the metabolism. It can be shorter, it will be replicated faster, and just as in the hypercycle, the system in the end would go extinct. Well, not so if you are surface bound. So imagine that there is a mineral surface, and the assumption is that all of the replicators, not the parasites, but all the other replicators, the different types, are necessary for metabolism to run in a certain limited neighborhood. The system is modeled as a cellular automaton, where the reactions can be modeled, and also the diffusion on the surface can be modeled according to established techniques. And it just shows that if this neighborhood um, is such that you have at least one copy of all the different type of replicators that are necessary for running the metabolism, then there is the possibility of replication or into an empty site. If the metabolism is not locally complete, so to speak, then it doesn't work. It does not happen. And there is also the replication neighborhood. So it has to be into an empty site, and we don't cheat. The replication rates of the different molecules can be different, and the probability that a new copy will occupy the empty space is proportional to two things: the efficiency of the metabolic neighborhood and the replication rate constant of the particular replicator. And you just run the system, on a grid which has a torus topology, so that you don't have these edge effects and so on. It's just following the rules of the trade. Now, what happens? Well, it's very surprising. What you will see is actually a robust coexistence of this stochastic competitive system. So you have protected polymorphism of competitive replicators, and there is a cost of commonness and advantage of rarity. Why is that so? So that means if you are a replicator and you have the highest density across the whole grid, then you have a good chance that you will see around yourself other copies of yourself. Now, if you see other copies of yourself, then you very quickly reach the limit, so that in the local neighborhood, metabolism is not complete because the other synergistic partner replicators cannot be there. Okay, and then you cannot replicate. The advantage of rarity for a replicator, which is a slow replicator, is that if it looks around, it's much more likely to see a complementing set of the unlinked members of the genome, as it were. And this is what you see. So you get a protected polymorphism, which means that the system fluctuates, of course, because it's stochastic, but you are not losing anything of the replicators despite the competition among the replicators. Interestingly, this coexistence does not depend on mesoscopic structures of any kind. It looks completely random. Parasites cannot drive the system to extinction. unless the neighborhood is too large. So it's actually the increase of the metabolic neighborhood that approaches the um, uh, phenomenon of the well-stirred system. Of course, I mean, if you increase the neighborhood, then all in the end, everybody will be present, and you will have to converge to the well-stirred system. Uh, we also analyze the interesting phenomenon when a parasite can evolve into one of the metabolic replicators so that it can catalyze metabolism. That's also possible. And it survives many kinds of perturbation. So it solves the coexistence problem, even though there is no higher level unit of evolution, just local interactions. But it's surprisingly robust. Now there is another model, and it's an adaptation of the so-called trade group model by David Sloan Wilson. So basically, here is an example. You have got two types of replicator, the blacks and the whites, and you have a mixed global pool, and uh, and you take a sampling into local groups, you random dispersal. So you have several local groups or teams, as it were, um, and they will have a statistical composition depending on the densities of the mixed global group and what certain people would call the co-infection group size, or how many particles are infecting any local patch. And then you let the interactions happen, and then you make a harvest, right, into the second generation of the mixed global pool. And again, you see, suppose that one of them is an altruist, the other one is a parasite. The interesting finding is that this can also solve the internal competition problem, but not any kind of uh, problem, and you will see what's necessary here. So why does the trade group work again? I mean, put in writing. It works only for cases when the red hair theorem applies. What does it mean? The red hair theorem in statistics says that the people with red hair overestimate the frequency of people with red hair in a population, and that hinges on the fact that they know about themselves that they have red hair. So what counts here basically is the average subjective frequency, given that you feel your own presence. So in terms of this altruistic talk, what is necessary here is the molecules must be able to scratch their own back. Well, what does it mean? In concreto, it means that a replicator catalyzing metabolism can consume the synthesized nucleotide for its own replication, right? So one molecule of a certain type, just one individual, can it feel its own presence, because it catalyzes metabolism. It can also reap the benefits of this catalysis. That's critically important, as you will see shortly on another system where this condition is not met. So this is another way to solve the coexistence problem with some limitations, with transient group formation. And this can be experimentally realized. Here is the paper that was dealing with it. So you have these water in oil droplets. This is the boundary of the droplet, and we are interested in whether parasites can kill a system that does something useful. So we are using RNA molecules that are replicated. Since we do not have a generalized replicase RNA, we are using the Q-beta RNA replicase. That's a protein, and that has to be given to the droplets. So not only the activated nucleotides for the replication, but also this component as well. And here you will see that the Q beta replicates this. Now there is this very interesting thing that in the middle you can actually paste this molecule that can do useful work. So that's the catalytic domain, which means there is a substrate, and if that catalytic domain cuts this substrate, which is also a polynucleotide, then you produce a fluorescent product. So the degree of fluorescence will tell you how intense the enzymatic aid to this replication, to this chemical reaction is in the particular droplet. Now, what you do, in fact, here you have the same image, is that you go through this cycle. So here is the global pool. Then you put randomly the molecules in the compartments, and you just throw out everything which is not fluorescent enough, and you go for several cycles, right? And here is the microfluidic machinery whereby you can do it, right? So those that light up nicely, this is a bifurcation in the microfluidic system. This is thrown away, and this is kept, and you go through this cycle. What is a parasite here? A parasite here is a replicating molecule that has lost the central part, which is the catalytic domain. If you have many of them, fluorescence is weak, okay? But then you will get thrown away. And it's transient, right? Again, so it's the global pool, the droplets, the global pool, droplets, they are alternating. And as the title says, this transient compartmentalization of RNA replicators prevents extinction due to parasites. Beautiful. Can you do better than that? We will see later. But I would like to give you a third example where actually it is true that molecules cannot scratch around back. So imagine that you have got a replicase RNA that can replicate different kinds of RNA molecules. And also itself, the question is what efficiency? Now, from the lesson that you have learned, it's obvious that if this arrow is stronger than this arrow, the parasites will take over without population structure. What happens is population structure. And this is the case where a replicase cannot scratch its own back. Why? Because a replicase enzyme, it's a template. So it's a second order system. So another copy of itself must be necessarily there, right? And then they can turn these two rows randomly. That basically leads to the observation that kin selection on the rocks is what you required. So if you are increasing the diffusion rate of the replicators, you are losing the high probability that you are, as an altruist, you are interacting with a copy of yourself, and the system goes down the drain. What we have been able to show that if we have got three traits of the RNA replicators, the target efficiency, the replicase efficiency, and the copying fidelity, and these are in a three-way trade-off with each other. So you can maximize only one for a particular molecule, and then the two others are going to suffer. And this is an emergent outcome in a replicating population. If it's again a cellular automaton model, if the diffusion rate is small enough, what you see is that the functionality of the different molecules is increasing with time, despite the fact that all kinds of parasites appear all the time. So the quasi-stationary population is always bimodal, but this peak is actually much higher. These are the parasites. The longer ones are the more or less efficient replicase molecules, right? And this is the summary. So as time passes, the average length of the molecules increase, the error rate of the molecules is selected against, it goes down, and the replicase activity is going up across the whole population. And again, this collapses without a kin selection, helping your own relatives due to limited diffusion. Now, that's about the multi-level selection of type 1. Here we go now to type 2, is reproducing compartments. Okay, so the molecules replicate and compartments also reproduce. It's much more efficient. First, I want to mention briefly that we have this phenomenon or category that I coined the infrabiological systems, where, so this is a living system, a minimal living system, which has all the components, method, boundary, and template. And any two produces an infrabiological system. Infrabiological because almost biological, but not quite. These are the different possible combinations. Here is one that was suggested by Tibor Ganti. We shall mention him later as well. So you have got kind of a protocell, and inside the protocell there is an autocatalytic metabolism. Here is the autocatalytic step. This is what it is consumed. This is what is product. But it also produces this membranogenic molecule. So basically, it's an autocatalytic metabolic cycle enclosed in a kind of a lipid bilayer. So that you have got metabolism and boundary, but no genetics. It's undoubtedly an autopoietic system. This is paper chemistry. Well, it's 1978. But this can also be realized. I mean, I don't want to go into this because there is no genetics into it, but it's so interesting. So this is actually the foremost reaction, which is autocatalytic. It's an autocatalytic production of sugar molecules that actually eat the molecule like glycolaldehyde, and these small molecular autocatalytic cycle is put into droplets, and you will see that only those droplets grow, into which at least one molecule of one member of the foremost reaction system is put in. And using microfluidics, if the growth reaches a limit, you can split the molecules into, the compartments into two halves. It's again in a water in oil system. It's an aided type of replication, but basically reproduction. But any kind of disturbance could actually give rise to the fission of the compartments that have grown beyond a certain size, and that has been demonstrated. Now, let's go now to the more interesting and still to be made conceptualization of a minimal organism here. And I think so far that's the best conceptualization that is deeply rooted in chemistry. So you have what you saw before, a metabolic engine, which is autocatalytic. It produces also the constituents of the replication subsystem and also the membrane subsystem. And these are also linked together. This can help actually to synchronize the growth. This model is called the chemoton, and all the subsystems are autocatalytic, and the system as a whole is also autocatalytic. The chemoton shows organizational closure. It is autopoietic, creating and maintaining its boundary conditions. It's deeply rooted in chemistry, yet presents an abstract and idealized model. No enzymes in the basic model, which is good. Enzymes are highly evolved things. Other catalysts are okay. In fact, if you actually go back to it, I mean, the metabolic engine is also a catalyst, right? It's operating in cycles and it produces different kinds of molecules. And unlike these molecular replicators, this is already a minimal organism, something that we would like to actually materialize in the coming few years. What is also important, that it has genetic control, but it doesn't have to be extremely complicated. And we have the bone wall from Maynard Smith that fortunately genes don't have to encode for the laws of physics and chemistry. So there's a great deal of self-organization also going on. And as Stu Kaufman would say, so the chemoton is in that sense a system which satisfies the definition of the Kantian wholes. And here you can see the last quote, especially important. The parts of the thing combine into the unity of a whole because they are reciprocally cause and effect of their form. Exactly. Kant, of course, did not know about molecular system, but it's actually very important that this is exactly what's happening in such systems. And now, going to the higher level of evolutionary unit, so a model of a system like this was created in 1987, a mathematical model. It's called the stochastic corrector model, in which you have got reproducing protocells. They grow inside the constituent genes are replicating, and once again the genes are replicating and contributing to a common metabolism. Now we don't cheat. We allow internal competition inside the protocells, but we find that despite this internal competition, at the protocell level, a stable population rises. How come? How is that possible? That is possible because during growth and also during division, there is stochasticity. We don't deal with concentrations here anymore. Because of the small numbers, we are dealing with numbers of molecules. So it's stochastic chemical kinetics that have to be applied. So you model independently reassorting genes. You select for optimal gene composition between the compartments, and competition among the genes within the same compartment is still allowed. And stochasticity in replication fission generates variation on which natural selection acts, and it actually acts successfully. So here you have got the replicators that are evolution units, and here the protocells are also evolution units because they actually reproduce. So a little bit of technical detail here. All the genes are independently reassorting upon fission. You can think of ribozymes in compartments, but of course it can be anything, any molecular system that satisfies the same criteria. You have selection for optimal gene composition between the compartments. Stochasticity in replication and fission generates variation on which natural selection acts, and the stationary compartment population emerges. This is something that you see again and again. For example, if you think of the eukaryotic cell, you will see that it's also a similar problem because the host cell engulfs the ancestor of mitochondria, then a certain lineage also engulfs the ancestor of plastids and so on. So it's a similar problem, and at the beginning it also gives the same problem. Okay, yeah, I mean, you have a very nice combination of functions here, but you have the problem that each member was an independent replicator. So how can you ensure that the higher level unit of evolution is not disrupted? It's exactly this type of selection mechanism that I have described. We have also analyzed very recently the transition between the surface-bound metabolic system that is then giving rise to this protocell-based stochastic corrector type of mechanism. We were interested how does that really work. It's a rather heavy mathematical modeling. The idea is that, as Günther Wächter once proposed, that you are producing this molecule on the surface, and there will be also molecules that can generate membranes, and then bubble-like structures not shown here emerge, and then what is called abstraction, they can be liberated from the surface. Of course, it has these constraints on the permeability of the forming membrane and so on. I don't want to deal with that. That's another subject. But we have shown that is actually a successful process, and there are some interesting, a bit technical but interesting conclusions one can draw. So here are a few findings. It's easier to initialize operational metabolic replicator communities on a surface than in vesicles. So the surface could actually be a stepping stone. Vesicles with a higher order of complexity can be initialized from replicators as evolved on the surface. I mean, that's the successful abstraction or transition. Replicators in the stochastic corrector model are less strictly selected for low degradation rates. Instead, they are optimized for faster replication. The stochastic corrector model is replication-driven, whereas the metabolically coupled replicator system on the surface is degradation-driven, meaning that you are selecting more for the resistance of spontaneous degradation than for replication rate. And the stochastic corrector model selects more strictly against high variance in replicability, as its replicators have to evolve similar replication rates to maximize the number of functional offspring vesicles, that is, to minimize the assortment load during vesicle fissions, right? I mean, if you have a strong internal competition, with very different replication rates, then when you make the stochastic fission, you run the risk that one or even both compartments do not have at least one copy of all the necessary genes, right? That's called reassortment. And there is very strong selection, as it was already shown decades ago. There is very strong selection for replication synchrony, which is actually not the case on the surface bound metabolic replicator system. So this is actually a case where you go from a multi-level selection of type 1 into multi-level selection type 2. It's really models the transition, a major transition if you wish, but in detail, right? Because there's a tendency to analyze the state of affairs before the transition and then after the transition, but it's not so common to analyze how, dynamically speaking, the transition itself occurs. But this is an example of that. Here is a helpful survey of several of the concepts I was discussing, a review from the 2020. So some of the things, especially last item, are not included there for obvious reasons, but it's a helpful survey, and you can actually look for many of the details here. And with the chemoton symbol, with the three qualitatively different autocatalytic systems linked to each other into a higher level whole, I would like to thank you for your attention, and I wish you a very successful meeting, and it's going to be very successful. I'm reasonably confident.